Tonight's topic and article is about 3' end of the 3' untranslated region, 3' end UTR of different isoforms of the mRNAs, which influences maybe by enhancers. And the important thing is here, we know that, from all people know that, enhancers can simulate two different things. Previously, we know that enhancers, most of the time, they are contribute to produce transcripts. Second thing is interesting one. Maybe in processing of this mRNA, they have an important role too. Yes, tonight they discovered in this article these enhancers and maybe some other transcription factors and other factors are stimulate and regulate, most of the time upregulate 3' end isoforms of the mRNAs. What we can see tonight is here. First, P10 gene, one of the famous genes that we know from our previous studies. We know that P10 is one of the most important inhibitor in a most famous pathway in cancer studies, RAS-MAP kinase pathway. And this P10 is the really important inhibitor. And we know that it has a role in different syndromes like Cowden syndrome. And we know that this protein is one of the really important multi-UTR gene that we know. Okay, in previous study, we know that this gene has 50% of the genes are single UTR. On the other hand, half of the genes are multi-UTR, and this is one of the famous ones. And its enhancer can have different roles. Imagine what we have in panel 1 is actually, by CRISPR gene editing with guide RNA, we can eliminate the enhancer of this gene. How happen? What would happen next? This is what we can see in the panel 1 of this article. We go further and see whether if this enhancer, check some point in this panel, check whether it can influence the measure and the amount of the mRNA transcripts or not. If it's not, actually it's not, because in multi-UTR genes, this enhancer most of the times, instead of working on the amount and number of transcript production, they work with transcripts processing more. And the interesting thing is here, we know that these enhancers most of the time work like turn on and off for single UTR genes. On the other hand, these enhancers, a lot of the time, for example in P10, they work for different short and long form of the UTR. We can see the result in panel 1 after this introduction. In panel 2, we wondered about if other genes can have same behavior or not. More precise work, if a promoter of genes like GAPDH or promoter of other genes and the binding protein to the promoter part and enhancer of the different genes can play a role instead of the enhancer of the P10 or not. And in panel 3, actually, we wondered about finding spectrum of different genes, like we find here this important gene, which we can find with GIPSEQ sequencing. The enhancer part, actually with H3K27 acetylated. Wait a minute. I guess you can think about these histone acetyl transferases maybe can play a role in different isoforms of 3' UTR of isoforms of the mRNA. Yes, this is right. At the end we can find it, but here in panel 3 we can find These style enhancers regulate CPA activity for different polyalylation cleavage. And we can find for NUDT21 gene. If we do it and see whether the amount of transcript and see whether the influence of enhancer can change something about this gene or not. See more. What about the transcription factors? What about the transcription elongation factors? We know that these can work, and these are part of the big machine. RNA polymerase II mediator as a key important part of the transcription factories. We have this notion right now in 2023. Transcription factories and polyadenylation polyadenylate polymerase and CPSF and CSTF, different parts of the polyadenylation complex together with polymerase II. These big machine and the CTD part of the polymerase II can influence by different factors, and they can, as a result, influence for specifically these multi-UTR gene, different isoforms of the 3' end of them or not. In panel 4, we can find interesting transcription and transcription elongation factors are widespread, regulate the CPA activity processing. As a result, there is a really important question. Most of these multi-UTR genes are condition-specific and, more importantly, tissue-specific genes. These enhancers and these different isoforms can stimulate, for example, in erythrocytes, while the simple differentiation and stem cells. We know that in the blood, we have a lot of them. Is these different isoforms can influence their fate at the end or not? We can find in panel 5 this discussion. And after all, we will have this discussion at the end.